We're all set for that. Let's jump into that phase of this stuff. Let's talk about mitosis and the cell cycle real quick. This is, of course, chapter 12. Just a small piece of it, though. And let's start off by mentioning and reviewing ourselves on the cell cycle, so we can go through that real quick. Really didn't matter, I guess. I want to draw a pretty circle today. Yay. Okay, very good. Cell cycle. Let's talk about this little small piece right over there. We'll divide this into two sections. Well, I've already ruined it. This is going to be M, which is standing for what? Okay, mitosis. And what's the C standing for? Cytokinesis. Mitosis means to split the DNA. Cytokinesis means to split the rest of the cell. And then we've got two other phases to wear ourselves with. Actually, three other phases. We've got G1. That's important. G2. S. And then G2. Gap 1. Synthesis phase, and then G2. And obviously the cell cycle has to be going in this direction that we're seeing right there. Cool, so there's our cell cycle, essentially. Awesome. What is G1 and S and G2 known as? Say what? Okay, so that's known as interphase. Write that up at the top. Just throw that in there. Make sure we got that. And then remember that G1 is basically a section or a time frame of normal growth. S is, of course, synthesis of DNA. And G2, of course, is prep for mitosis. I want to talk about a couple of checkpoints, and I really want to spend some time talking about the checkpoint that we see right through there, the G1 checkpoint, and also the G2 checkpoint. So in the G1 checkpoint that we're seeing right there, G1 checkpoint is basically the primary checkpoint, because passing the G1 checkpoint means that you're probably going to finish the rest of the cycle. So G1 checkpoint essentially causes the rest of the cycle to complete. Remember, if you don't pass G1, if you don't go through it, then you basically go into the G non-dividing stage. And then we have to talk about some of the regulations. So let me go over that real quick, because to me that's something else that's really important to understand. You've got this molecule called CDK that is consistently in your cells and around your cells. Do you remember what CDK stands for? Okay, cyclin-dependent kinase. And your CDK levels are consistent all the time. Now, once you are in this prep for mitosis phase and you're getting ready to jump into mitosis, your CDK needs to have combined with a molecule called cyclin. So right here, you're seeing my CDK that is mixed up with a little baby cyclin, and they fit together perfectly. Cyclin is the regulatory molecule because cyclin is the one that's not always present. And what is this duo called? Do you remember? Okay, it's called MPF, which stands for maturation promoting factor. I always remember that it's basically like the mitosis promoting factor, but it actually is a maturation promoting factor. Cool. And so once you have MPF, you can go through and get into mitosis. Now what happens after you've entered into mitosis and you've gone through cytokinesis is your CDK unbinds itself from the cyclin. Now there's your CDK, normal, ready in its chill spot, ready to recombine. And then there's this little broken up cyclin right there. Now that cyclin's broken up, the cell cycle is not going to continue onward. Makes sense? So in short, what you're seeing from this, kind of the most important piece to see is that cyclin if you will, regulates, at least in some part. It's not the only regulation, but it regulates in some part the cell cycle. I think the last little thing that I want to get through in this section is going over some other just unique vocab. And I actually have done this in some prior videos as well, but I'll go through this. I want to go through the terms glucagon, glycine, glycerol, guanine, glycogen, glucose, fructose, fatty acid, phospholipid, and insulin. So some of these just vocab words that I want to go through. It seems pretty random that I choose them, but it's all good. Glucagon is going to be pancreatic production, a pancreatic hormone that puts away, I'm sorry, doesn't put away, that causes or stimulates glycogen breakdown. Glycine is just an amino acid, probably actually the simplest one. It's a simple amino acid for sure. Glycerol, that is the head to which fatty acids bind in a fat, and I'll show you that in just a second. Guanine, like as in C to G and then T to A, guanine is the G there. It is the it's a DNA base and also an RNA base because it's in both of them. Glycogen, of course, that is the glucose polymer. In animal, I almost misspelled animal. I did misspell animal. In animal tissue. That's how we put away glucose in our tissues. Glucose, of course, is the most basic monosaccharide. Fructose is another monosaccharide. Remember that this one's not as easily broken up as glucose is. The relationship between those two, though, I think we want to know, is being isomers. The same formula but different structures. A fatty acid, of course, is what combines with the glycerol. Combines to glycerol. It usually contains many carbon and hydrogens. So really it looks like that. If you want to see a picture of it. There's your fatty acids right there. And there's your glycerol head. You want to draw that little picture in your notes. Phospholipid, of course, is something like this, but this is found in cell membranes. Remember, it's got both hydrophobic and hydrophilic portions. Both hydrophilic and hydrophobic. And there's insulin. And the purpose of insulin is basically to lower blood sugar, which of course would be high post eating. So there we go with all that.